medicine. Bill.
Thanks, Roy, and thanks to the organizers. Someone said something like, everything that's been said on this particular topic has been said. It's just not everyone has said it. So I want to thank Lowell Schnipper for giving me some leeway here to talk about something a little different. And what this slide is showing on the right-hand side is the Gordian Knot, or at least someone's idea of what it looked like. And as many of you know, the legend that Alexander the Great was shown a chariot that was tied to a pole by this intricate knot, and the idea was the person who untied the knot, and only that person, would be the future conqueror of Asia. And unfortunately, Alexander could not find the origins of the knot or the origins of the problem to get at the knot, so he took out his sword and he sliced through the knot. And the phrase cutting the Gordian knot has come to denote a bold solution to a complicated problem, but perhaps it would have been better to have prevented this knot from being tied in the first place. So assuming that there's something to that, let me tell you what I was thinking about, and perhaps I woke up on the pessimistic side of the bed when I was putting this together. So here it is. Despite substantial progress, and that has been really substantial, most established cancers remain relatively impervious to treatment. I hate to say it. Chemotherapy, radiation, and maybe immunotherapy are our swords, and perhaps targeted therapy are daggers. So I want to make a prediction that improved diagnostics combined with informatics and all the things you heard about systems biology will allow robust predictions of cancer susceptibilities in the identification of premalignancies or cancer susceptibility disorders. And ultimately, we might move from a society dedicated to disease care to one more focused on health care. So this is a slide from Bob Weinberg's book. You're all familiar with the idea, probably beginning with Bert Vogelstein, that it takes many, many years to go from a phenotypically normal and maybe even a relatively genotypically stable epithelium through a series of alterations that lead to first the genotypic changes of premalignancies to ultimately phenotypic changes that you can identify as pre-cancers or pre-malignancies. And there's many good ones. And there is certainly the possibility, and there's examples, that during these early steps there are alterations that are much more amenable to relatively simple therapeutic approaches, which would now be called therapies of a pre-malignant condition, than when the disease is well established. So for example, an aspirin is highly effective in preventing colon cancer, and probably an aspirin or other COX-2 more specific inhibitors are very good at treating adenomatous polyps. From most of your experience taking care of patients, you give an aspirin or Celebrex to a patient with colon cancer, and perhaps the only thing that might happen is you'll make them bleed. There are a variety of these pre-malignant conditions that have the potential for us to get at. Razel made an excellent point that chronic myelogenous leukemia in many ways is the initiating event, is the chronic phase really a premalignant condition driven by a single genetic driver, and that's why imatinib is so incredibly effective, prolonging patients' lives very, very well. But if you get to the end of the line with blast crisis and genomic instability, where all hell breaks loose, this type of approach is a lot less effective. So let's see. So I want to give just one example about a premalignant condition that has caught my attention over the years, and that's Smouldering myeloma. My commercial colleagues told me, well, no, there's no commercial value in Smouldering myeloma because very few patients have Smouldering myeloma. I point out to them that, well, yes, every patient who has myeloma had Smouldering myeloma at one point. They just were never diagnosed, and they have taken a slightly different view. But here's a trial that comes from Celgene, sponsored trial of lenalidomide plus dexamethasone in patients who were identified as high risk by Matteo et al. And these patients had a relatively predictable onset to myeloma, which of course is to symptomatic myeloma, which of course is a fatal disease. So the standard of care for smoldering myeloma is observation. So in this particular trial, they randomized 119 of these high-risk patients to the treatment arm versus observation, which was the standard of care. The treatment was lenalidomide plus dex, and then a maintenance program. The primary endpoint was time to symptomatic multiple myeloma, and there were secondary endpoints, very small, underpowered trial, lots of provisos, response rate, overall survival, and safety. The results, in my mind, were relatively predictable. For time to progression, the median had not been reached at the time of publication. It was 21 months for the observation arm with a hazard ratio of 0.18. In this very small trial already, there were indications of improvements of overall survival. The treatment group at 3 years, 94% were surviving versus 80% on the observation, a hazard ratio of 0.3. But if you look from the time of diagnosis to smoldering myeloma at 5 years, 94% of the patients were doing well on the treatment arm versus only 78%, a hazard ratio of 0.28. And although the treatment is not without side effects, and it's one of the very first interventions, I think directionally it's quite interesting. So are there policy implications to any of this? Maybe. I would put out there that a policy that promoted an increased understanding of the basis for and diagnosis of genetic susceptibility and the movement to phenotypic progression of disease, that would be an investment, I think that would be very, very important, and someone raised it earlier in the discussion. I think policies that help identify and validated surrogate endpoints of clinical benefit would be critical for this. Otherwise, the trials will be costly and perhaps not feasible. And again, there are policies working, again, together with regulatory colleagues to help shape the regulatory environment to encourage these sorts of early interventions. I think that's it.